Good morning, good afternoon, good evening. Welcome to the 8th South Forum on Sustainability. The theme is the politics of hope, people's resilience and resistance in catastrophic times. Today, we have a lecture on the loss of biodiversity, an insect perspective by Professor Chensheng Alex. We provide English, Chinese, and Spanish simultaneous interpretation. You can choose the interpretation at the bottom of the computer screen. We would like to thank today's interpreters, Lola Laventry, Mercedes Pico, Liang Yin, and Li Mengheng. May I first introduce the co-moderators for today's section. My name is Xue Cui, Margaret Jadi. I am Associate Professor at the Institute of Rural Reconstruction Movement of China, Southwest University in China. I am board member of Asian Regional Exchange for New Alternatives and one of the founder members of Global University for Sustainability. I have been actively involved in the rural reconstruction movement in China since 2000. Professor Deng Zhishan Denise is teaching culture studies at Lingnan University. She is Deputy Director of Master Program of Culture Studies. She is also Acting Director of the Center for Cultural Research and Development. Professor Deng is an interdisciplinary ethnographer who focuses on inter-Asia gender and sexualities. She is the author of Conditional Space, Hong Kong Lesbian Desires and Everyday Life. Her articles were published in the Sociological Review, Sexualities, Gender, Place and Culture, Media, Culture and Society, and GLQ. Thank you very much. Professor Deng, please.
Thank you, Margaret, for the introduction. I would like to introduce, of course, with honor, Professor Liu Chensheng, who is here to present a talk today on the loss of biodiversity from an insect perspective, which the title itself is already very attractive and timely for us. So Professor Liu's research focuses on assessing pesticide exposure and its toxicological effects in ecological and human health. Before being appointed as the distinguished professor at Southwest University in Chongqing, China in May 2018, Professor Liu was on the faculty at the Harvard T.H. Chan School of Public Health in Boston, Massachusetts, USA from 2008 to 2018. Before that, Professor Liu was an assistant professor at Rollins School of Public Health at Emory University from 2004 to 2008. Professor Liu received his PhD degree from the School of Public Health at the University of Washington, Seattle in 1996. Other than these academic appointments, of course, Professor Liu have served as an associate editor for Environmental Health Perspectives between 2006 and 2016, and on several committees at the federal and state levels, including the Scientific Advisory Panel to the US government's Environmental Protection Agency, under the authority of the federal insecticides, fungicides, and rodenticides since 2004, and the National Academy of Science since 2013. Professor Liu is currently an adjunct professor at Harvard T.H. Chan School of Public Health, the University of Washington, and Tufts University. If you have the opportunities to search, actually just even briefly on Google, you'll find all the impressive work that Professor Liu has been doing and still doing right now on this issue, especially on the focus of honeybee colony collapse disorder, which is talking really about the everyday, effect of harmful this is a difficult word to pronounce, neonicotinoids in our environment and its association to the colony collapse disorder. So with privilege that we have at the South Forum, I'm here to present Professor Liu's presentation to all of us now. Thank you.
This is also my honor to participate in this forum. I think, my research, although has been focusing on pesticide exposure and human health, by no means I am I'm the ecologist that dealing with a lot of insect. However, a television program way back in 2005 actually sparked my interest in this association between anthropogenic activity and the loss of one insect. I think most of the people agree that we are losing many organisms, in part mostly because of the change of environment, as big as polar bear because of the climate change, or as small as the honeybee, right? So without this horrifying disease called colony club disorder, many of us, including myself, would not be aware that this tiny insect called bee or honeybee are losing their population size because of a new pesticide when was introduced in the early 1990s. They claim that this type of pesticides will revolutionize the use of pesticides and will improve the safety of pesticide to the ecology, in particularly large mammals. However, this disease we call colony club disorder actually changed the tone in a very dramatic manner. I was sitting in my living room way back in 2007 when I was watching the CBS 60 Minutes in my very comfortable living rooms. And all of a sudden, this gentleman called David Hackenberg was brought into the camera, and he started talking about his career as a commercial beekeepers and a symptom or a situation that he has never encountered, which is now we call colony club disorders. So exactly what was the problem way back in 2005 in the winter when Mr. Hackenberg parked his 4,000 beehive in southern Florida, so the bee can be comfortably overwinter there. He said this, for my 40 years as a commercial beekeeper, I never witnessed this phenomenon in which more than 90% of his 4,000 beehive lost bee over almost like a 10 days period. When he said lost, he meant that no dead bee can be found nearby the hive. It's not because the bee were dead, or he can find millions, if not tens of millions of the dead bees. He described the situation as bee disappeared. So just like other people, when they witness something that's very weird and strange, he picked up the phone and called USDA, which is Department of Agriculture, and have those bee experts flew to southern Florida and to inspect his beehive. And at the end of the inspection, everybody agreed that they haven't seen this type of a problem before. That's why they gave this name, colony club disorder, to describe this heartbroken and huge disaster event. So before we get into further, I believe that most of the audience here tonight probably does not have a lot of knowledge about bees. So let's spend a couple minutes to go over what are those bees. So bees actually came to this planet, this Earth, much earlier than human beings. The primary species in our study is Apis mellifera. This probably the most prominent bee on Earth. In China, there is a relative to Apis mellifera called Apis mellifera. And I believe that this species is very unique to China. So bee, unlike other pollinators, bee is probably the only pollinator that is perennial pollinator. What does that mean? Meaning that the bee, as a colony, they can over the winter on site over and over again, as long as the hive can safely and successfully overwinter. Other type of pollinator, they either had to migrate to the south, like the most famous monarch butterfly. When they migrate in a large population from California to Mexico every fall, they create a beautiful ecological phenomenon because they cannot sustain the cold winter in California or further north. But honeybee can stay on site over winter. So we know that each hive has one queen, has about hundreds of drones, which is male, and the rest of the worker bees. Those worker bees are the daughters of the queen, right? Obvious. The queen herself only left her colony once in her life. And we call this travel maiden flight. This is where the word came from. So the bee, the queen, flew vertically on top of her hive about 40 meters. And she actually brought every single drone bees so they can have mate. And queen suck up the sperm for as much as she needs for the rest of her life. And then she went back to the hive. And after that, queen never came out again. So for the healthy Italian bee or Apis mellifluus, there should be 40 to 60,000 bee per colony, depends upon the location, the weather, and so on and so forth. But 40 to 60,000 bee is a good estimate of the size of the colonies. So for the whole colony to be able to successfully overwinter, the beehive as a whole, they have to work pretty hard to generate heat. Otherwise, they couldn't maintain the ideal or critical temperature inside the hive. And most of the energy actually generate from the thoracic muscles, where the wings and the legs are attached to that part of the bees. What is very interesting to our study and probably to this colony collapse disorder is that bee has different life cycles. This sounds maybe shocking or fascinating, but this is true. For the queen herself, a healthy queen, she can live somewhere between 3 to 5 years. For the male drone, they only live from spring to the early or middle fall. By the time before the winter arrived, male drone got kicked out by the workers and they die outside because of cold winter. And for those worker bee in the hive, even though they are the daughter to the queen, they have dramatic different life cycles. For the spring, summer, and the early fall worker bees, their life cycle somewhere around 30 to 40 days. Nobody lives longer than 40 days. Now that's the fact. However, for the last generation of the year, which means that for the last batch of eggs that the queen lay, those bees, we call them winter bee. They can live as long as 200 days. So in other words, those worker bees that emerge from late fall, they can live, their life cycle is much, much longer than their sisters from the same years. And this actually is very important to CCD. And this is very important to our studies that I'm going to introduce you later. For most of the scientists who is engaging in learning how to live longer, for example, extending human life beyond 100 years, they all look into honeybee models because they want to know why the daughters have different life cycles and mother also has much longer cycles. So if human being can identify or find out what really controlling different life cycle in this species called Apis mellifera, then most likely human life cycle will be much longer than what we have right now. But so far, nobody can crack this secret. And this is part of a reason I believe that bee is a highly intelligent insect, probably the next to human beings. What I'm going to say here is that what I just list here on this slide are information that is critical to our study and also critical to how we're going to study colony collapse disorder. Bee has other fascinating characters that will probably take months to introduce to everybody. So how important is honeybees? They pollinate about 100 different crops. And those crops are very important to sustain human lives from the nutrition purpose, from the consumption of the food purpose. So as I described earlier, that when those experts, the bee expert arrived in Florida, they also agree that they have never seen this type of bee deaths before, because in most cases, when a hive fail or dead, you can find thousands and thousands of dead bees, either inside at the bottom of the hive or scattered around near the hive. But in this case, they can only find very, very few dead bees. So they give this name colony collapse disorder. And CCD basically is a symptomatic disease. There's no way that you can find bees or find those dead bees to identify the problem in terms of either bacterial infection or mite infections or so on and so forth. You can only identify either this hive is that of CCD or something else by the symptoms. So what are those important symptoms? For example, for those hives that actually just lost during the winter or after the winter, they were actually fine in the summer or early fall. In other words, Mr. Hackenberg said this: when we arrive Florida, I inspect every single hive. Every hive looked healthy to him. And that's why he believed that he has a very good year. And he's gonna take the vacation and then come back next year to start his cycle again. So his hive were healthy from the commercial beekeeper's perspective. However, after weeks, late winter or early spring, he lost 93% of the 4,000 hive. Also important in terms of identifying the CCD colony is that inside the hive, there are many, many left behind honeys, nectars, and pollens. Also, what's left behind were various stages of brood. In other words, those bees left the hive not because of lack of food for the winter. In fact, the queen were about to lay egg because the winter was about to over and then she started laying eggs. It's very, very uncommon for the worker bee to leave eggs behind without taking care. This is against the mother nature of honeybee, but it happened in those CCD hive. Another very strange phenomenon is that if you have an empty hive with honeys, nectar, and pollen inside, very soon there are other bees will come in and call this hive home. But in this case, for those empty colony CCD hive, There's just no honeybees willing to come inside the hive and call this place home. This is very, very uncommon to the to the honeybees. So what happened is that for most of the CCD problems, it did not happen to just one or two hives. It happened to many, many, many hives, like for example, 93. So if a beekeeper can lose 90 or 95% of his hive, it's gonna be affecting his pollination in the coming years. And this is gonna affect the agricultural production as well. If you go ask blueberry grower, strawberry grower, apple grower, and so on and so forth, those fruit trees that require significant pollination, you ask them about how is your pollination going for the last 5-10 years. There probably only one universal answer to this question, and which is that there is simply not enough bees to pollinate. And this is caused by the colony collapse disorder, which is very obvious. So it's becoming a worldwide phenomenon. For those audience tonight who understood or familiar with the agricultural production, they probably know that some of the pollination now is relying on bumblebee, not honeybees. Like for example, in Japan, there's many farmers. They bought a package of bumblebee from overseas company to pollinate their crop in Japan. Why? Because there is no enough honeybees to pollinate. So a couple of years after the discovery of CCD, United Nations actually come up with a publication, obviously asking us to change behavior to save bees, because bee is vital for food productions. Well, think about this. What type of behavior human has to change in order to save bees? When you take a hike in the mountains or some rural places, people will not get close to the hive because most of people are scared of hive, right? So what type of a behavior that human has to change in order to save bees? This is something I found very disappointing to the United Nations, because they don't want to say it clear and loud. They actually leave a very vague statement called changing human behavior. I mean, I believe that they know the causes of the bee losses, but instead of pinpoint to the exact cause of the most likely causes, they actually give us a very vague statement, say changing human behavior. So this is something I found very disappointed for the very important organization in order to save this very important insect or pollinators, but they are not willing to speak to the point, to the real cause of the problem. Instead, they say changing human behavior. So what would be the direct consequences of not having bees? Well, we probably won't have those beautiful, delicious fruit and vegetables. This is what's going to happen. Or this is what happened in China right now, northern part of China. Those village women, they hand pollinating the pear trees. They have no other choices but using their hands. I actually visited a couple of orchards in southern part of China and part of Taiwan. They don't have a huge population in the village that they can function human population, hand pollination. So what they were doing is, to many people feel disgusting, was to use the fly that we found is a nuisance insect. So what they were doing is to hang those rotten chickens on the limb of the fruit tree, and then those rotten chickens will attract thousands and thousands, if not millions, of flies. And by doing so, the fly actually work on this, I call it random pollination. And once the pollination is done, I asked one of the orchardists, so what are you going to do with all those flies? He said, well, I just spray insecticides because the fly won't go away. So this is a consequence, which is very shocking and disheartening. But if the UN people would be in the orchard with me, I asked them what kind of a human behavior has to change to save those bees. So as I mentioned, that Japan, since 2015, the whole country suffer from the loss of honeybees. So they have to import bumblebees from Israel in order to sustain the agriculture in Japan. And we will come to this subject later. So there are many causes to CCD at the beginning of these problems. For instance, the migration, the long migration, the frequent long migration of the hive probably most likely causes CCD. What they're talking about here is that, for example, Mr. Hackenberg, even though he parked his hive in southern Florida in the winters, by the time that the spring arrived in the south of the United States, he started put his hive on the trailer, an 18-wheel trailer, and he's gonna travel all over the places in the United States to pollinate from the south to the north. And by the time that he finished the last pollination, most likely in the northeastern part of the United States, and then he will come back to Florida to overwinter. So people suggest that this type of a migration, taking hive travel around the country, is likely the cause of CCD. And also because of lack of diverse food for the bee is another reason. And obviously global climate change could be the culprit to CCD. I mean, climate change here play a role in terms of messing up the timing of flowering for many fruit trees, for example. And this actually becoming often event which bothers beekeeper a lot. Another major reason that always stay with honeybee hive is pathogen infestations. There are many pathogens, mite, parasites, viruses that always stay with beehive. And this is why beekeeping is becoming a very labor demanding job, because beekeepers have to frequently inspect the hive. And once he spot the problem, then he has to use many ways to get the hive better and healthier. The last possible causes of the CCD is pesticides. But the pesticide here is referred to as acute mortality, acute toxicities, meaning that when the orchard is being sprayed by the pesticides, if you happen to let the hive fly around the orchard, then most of the bee will be killed by the pesticide in the very acute manner. But at the time when I look into this problem, I don't believe any of the causes, suggested causes, is related to CCD, because if you look at the acute mortality caused by pesticide, if you look into this pathogen infestation and so on and so forth, when a hive die, you will see thousands and thousands of dead bees inside or outside the hive. But CCD symptoms do not include this. And that's why I believe there are something else that kill or make the bee disappear from the hive. Now, I think this hypothesis makes sense, because as a physician or pathologist, when you look at the dead organism, you basically look into how this organism dead before you conclude what caused of the death, right? So if the CCD symptom is the emptiness of the hive, then none of the above suggested causes is relevant to CCDs. So at the time, I came up with my own hypothesis, which is the colony collapse disorder may have something to do with this type of the insecticide called neonicotinoids. This is a very difficult pronounced name of the pesticide. Even the native tongue of English people, they cannot spell this name well. But let's just call it neonics, okay? Neonic actually started being widely used in early 1990s, probably taking over the partial ban of organophosphate, which is another very popular insecticide. So in United States, for example, some of the organophosphate were banned in the early 1990s. So neonicotinoid fill in the void of this pesticide market very well. And it didn't take too long to let neonicotinoid become the most popular in terms of the frequency of use and the quantity of the use of insecticide worldwide. The reason is because those characters of neonicotinoids. First of all, it's highly water soluble. Most of the pesticide, either insecticide or herbicides, they are not very water soluble. So they have to rely on some other surfactants. But neonicotinoid is very, very water soluble. And that's why neonicotinoid also very systematic. What does that mean? Systematic meaning that this pesticide once applied, doesn't matter how you apply those neonicotinoids, it can be absorbed by the root of the plant or by the leaf or the stem of the plant themselves. And once the neonicotinoid get into the central circulation system, it actually, the neonicotinoids is distributed to every single cells of the plant. So from the plant protection perspective, this type of application makes sense because once an insect take a bite on any part of the plant, the insect got killed because the residues of neonicotinoid. And this is why it quickly become the most popular insecticides. But from the public health perspective, the plant actually, for example, vegetable or fruit, once it's harvested and then and was consumed by individuals just like the insect, we take a bite on something that has a residue of this systemic neonicotinoids. And unfortunately, those neonicotinoid poses neurological toxicities. And this is how they kill the insect. And in the meantime, when we consume vegetables and fruit that has some residues of neonicotinoid, we also consume some neurological toxicity of those pesticides. So at the time, I believe that because of the systemic nature or characters, it not only affect honeybee to the great extent, it would also affect the ecological environment to the same degree, just because those pesticides can come out as part of the pollen or can be taken away by honeybee as part of the nectar. This is how this very popular neonicotinoid called imidacloprid, if you can follow the title here. Imidacloprid probably was the first neonicotinoid being manufacturers in the world way back in 2003. When you look at the map of the United States, the color represent the intensity of the color represent the uses. If you look at this map, you come to a conclusion that most of the agricultural region use a little bit of imidacloprid. That's 2003. In the same year, when the CCD occurred in southern Florida, look at the use of imidacloprid comparing to 2003. Not only the area is wider, the amount of imidacloprid used more. 2011, even more so. 2014, not only every single agricultural region that uses imidacloprid, the amount of the usage are substantially increased comparing to 2003, right? Look at the type of the plant that use imidacloprid. It's very diverse, from pasture or hay, orchard, grape, rice, vegetable, cotton, wheat, soybean, and so on and so forth. Imidacloprid didn't, they were not using imidacloprid in corn until 2004. And guess what? In 2005, we have CCD problem. So timing is quite coincident. Before introducing to corn, we were actually fine without CCD. But 1 year after we introduced imidacloprid to the corn planting, we have this problem. Look at it right now. Very few imidacloprid used in corn, but soybean took over as a primary usage of imidacloprid from 1992 to 2014. Another very common neonicotinoid now in 2020 is chlorothalidone. This is the second generation of neonicotinoid. In 2003, it was not invented yet or manufactured yet. So nobody used chlorothalidone in the United States. But when this neonicotinoid got invented and manufactured, everywhere we use it. Pay attention to this dark red area. This is a Great Lake area, right? This is the barn of the United States. It's heavily agricultural regions. They plant mostly soybean and corn, probably nothing else. So that kind of gives you a sense that neonicotinoid doesn't matter if it's imidacloprid or chlorothalidone. They are associated with corn and soybeans. 2011, 2014. So it's the same type of a scenario to imidacloprid, that we use a lot of neonicotinoid in those agricultural regions. So unlike imidacloprid, chlorothalidone is primarily used in corn. And we know that in the United States, most of the corn are genetic modified corn. So it's very easy to attach neonicotinoid to genetic modified corn. And this actually very important because it actually set a tone for our initial, the very first honeybee studies way back in 2012, when we started thinking about how we're gonna find out exactly what causes CCDs. Our objective is very simple. We want to replicate CCD in situ settings, meaning that we don't get honeybee dosage in the lab. We don't look at individual honeybees. We look at colony as a whole, and we're gonna live this colony in the environment where we can start this dosing studies. So again, our initial objective is to replicate CCD. At the time, I didn't know we can be successful because it's looked like a very complicated phenomenon. So based on what we know about genetic modified corn, we know the usage of neonicotinoid in planting corn and soybeans. We come up with this hypothesis or study design. If you don't have enough knowledge of how pesticides are used in the United States, and if you don't know much about colony cap disorder, you may find this hypothesis unthinkable. I mean, how could you link genetic modified corn to this type of sugar water and then to CCD? But let me go through one stage at a time, and hopefully you will find this hypothesis very interesting and shocking, if you will. So, as I mentioned, most of the corn, especially big, large area planting corns, nowadays are genetic modified. Set aside the genes that being introduced to the corn, the manufacturer of the genetic modified corn companies, they also treat those corn with neonicotinoid before planting. What does that mean? If you look at these colorful pictures, the corn seed in the middle are the corn seed that our grandfather or grandfather would understand. The pale yellow color corn seeds, right? Those are the corn that we ate on the lunch or dinner tables. But to the left or to the right, those are the late 20th century, early 21st century corn seeds. They are genetic modified corn seeds, but coated with a layer of systemic neonicotinoid. Different color refer to different type of neonicotinoids. So why they do that? Because of their systemic natures. As I mentioned, once you drop a corn seed, doesn't matter is a blue color or red color, onto the soil, the corn grow, right? As long as when the corn grows, the residue of those red or blue color will grow as the corn. So as I mentioned earlier, that when an insect take a bite on either the leaf of the corn or the stem, the insect got killed, right? So when the corn grow like this tall, obviously there's going to be some residue left in the corn that we're going to eat, or it's going to be in the tassels or the pollens. When the wind blows, then the pollen, which contain a little bit of residues, would travel to some long distance. So what are we going to do with those genetic modified corn? Well, they're not going straight into the human consumption food chain, that's for sure. But we use those corn to feed the cattle, the chickens, the pig, and so on and so forth. And we still have left tons of corn unused. So we come up with distillation methods to turn corn into this fascinating sugar water we call high fructose corn syrup, or HFCS. Most of people would not know what HFCS about, but in the 1990s or early 2000s, every single sugar carbonated drinks, juices, or things that require sugar, pretty much you can find HFCS in it. This type of sugar basically took over sugarcane market. Why? Because of cost. The cost is very low. It comes with a liquid state, not a solid state. It comes with tasteless and colorless. So for the food production industry, they love this type of sugar water, right? So once they finish the distillation, they send those sugar water to everywhere by this semi truck, either to the bakery, to the carbonated drink company, and so on and so forth. And they still have a lot of sugar water left in the market. So, I mean, long time ago, beekeeper would use this type of water, sugar water, to replace honey. After all, honey is much more expensive in the consumer market, right? So the beekeeper would take the honey and to the market for sale. Instead, they put those sugar water as hydrocarbon nutrition back to the hive. So over the years, commercial beekeepers have used this. They admit that, and they also admit that this type of sugar water make the bee colony weak, but not as weak as it would trigger CCD. It didn't happen until 2005, 2006, when we start using those colorful corn seed to plant, and that started a problem. So the picture here at the bottom of the screen is the study hypothesis of Harvard CCD studies. We have 4 apiary locations, and they separate each other from at least 5 kilometers. So we don't have the interference between the beehive. On each location, we have 5 hives. 1 is the control. The other 4 are hives that treated with a little bit of neonicotinoid at different concentrations. So this is a double-blind studies. The beekeeper that help us to manage the hive, they didn't know which one is which. And this is part of the reason that we instrument this. So how popular is those seed-treated corn that come with neonicotinoid? In China, you can actually buy those corn seeds in those famous Taobao Wang. It's very difficult to find the traditional untreated corn seed nowadays, both in China and United States. So this is the picture of our studies. We launched this Harvard CCD study in 2010, and 2012 was a year that we have the first study's result. So what we did, go back to the screen here. So what we did here is very simple. We mixed the sugar water with a little bit of the neonicotinoid that we know the concentration. And then we gave those hive those sugar water every week for 13 consecutive weeks. January started from the mid-June and then to the beginning of September, because those apiary were set up in Massachusetts, which we have early, long, and very cold winters. So by September, we are getting those hives ready to overwinter, meaning that we are checking this pathogen infestation and so on and so forth. So we took over, we get those sugar water out of the hive, meaning the dosing was over in the early September, and we're getting ready for those hives to go over the winter. So what happened is that during the course of the dosing, and also including 12 weeks after the end of the dosing, we didn't see any hive has any problem in terms of the mortality. It doesn't matter is the highest concentration or the lowest concentration. The control also fine. So at the time, we thought that this experiment failed because 12 weeks after the end of the pesticide dosing, no hive have any problems. They're still healthy, they're still happy. But something magically happened 2 weeks later. We lost 3 out of the 16 hives that we treated with neonicotinoid. Another 2 weeks, we lost 2 hives. Another 2 weeks, we lost another 4 hives. And then more hive loss. At the end of the studies, we lost 15 of the 16 hives that we gave them a little bit of the neonicotinoid in the sugar water. So look at the pictures here. The picture were taken on July 19, 2010. This is a very healthy hive. According to basis on the amount of the sealed honeys, the amount of adult bee, the amount of the pollen, the amount of the brood, and so on and so forth. Every single competent beekeeper would consider this hive is strong, healthy, and so on. But this is also the hive that is taken sugar water that we added with a little bit pesticide onto it. The left-hand side of the picture, if you look at the top of the other beehive, the empty jar, that is the jar that we gave them sugar water with pesticide. So just keep in mind that this was a very healthy hive way back in July, but this was also the hive that is taking some pesticide through the sugar water. This was the hive that's controlled. You can see that this hive was successfully over winter. The 2010, 2011 winter in Massachusetts was quite long, very cold. So in early March 2011, you still see snow on the ground. But this hive was successfully over winter, but they exhaust all the honey inside the hive. So we have to feed them with some table sugar. Otherwise, they will be dead because of the hunger. So if you look at the left-hand side of the pictures, we put some sugar on top of the frame, so the bee can come out and then take it as a food. So here comes the classic CCD colonies. This colony was the picture of in July when we call the hive healthy. But in March next year, the healthy hive become like this. So picture here actually fulfill the definition of the symptomatic CCDs. You have left honey on your right hand side. You see there's tons of sealed or capped honeys. There are some pollens and so on, but you don't see bees either alive or dead. Look at the left hand side pictures. At the bottom of the frame of the hive, you don't see lots of dead bees. You only see maybe less than 100 dead bees. So by definition, this is colony collapse disorder hive. So remember that we have 4 locations of apiary. We lost 15. The only one actually survived in a very strange manner. This hive didn't live long enough, and they also collapsed. We lost 1 control hive, 1 out of 4 control hive. This is the control hive that died of disease, most likely pathogen infestations. And the mortality of this hive is dramatically different to the colony collapse disorder, because now you can see a lot of fecal matters. You can also see a lot of dead bees inside and outside the hive, which is very different. So when we first published these papers, obviously we were echoed by many beekeepers. They say, oh, we actually suspect the pesticide would be the cause of these problems. But we also face a lot of criticism. And obviously, most of the criticism comes from the pesticide industry. And those professors, they are sponsored by the pesticide industries. So the most loudest and probably the most critical argument was, they say the Harvard studies led by Dr. Liu was far beyond realities, because they used the dosage, which is 400 microgram per kilogram of high fructose corn syrup per week. This is way too high. The honeybees would have no chance to be exposed in their foraging environment. So they say 400 is too high. So we wait a while until all the feedback, either positive or negative feedback, came in. And then we make a explanation, if you will. So if we look at this 400 microgram per kilogram per week, this is the 100% correct numbers. There's nothing that's guessing or misleading or estimation. This is exactly what we put into the sugar water, okay? So most of people will understand the unit of this number. So each week, in each kilometer of high fructose corn syrup, there is 400 microliter of pesticides. This is what we put in the hive. So if you divide it by 7 days per week, 57 become the daily dosage. Assuming at the time when we were giving those sugar water to the colonies, there will be 50,000 bees in each hive. So if you divide it by 50,000, this is 2.8 nanogram per bee per day would be the individual's bee daily dosage. And assuming that those individual bee only live no longer than 6 weeks or 42 days, okay? This is actually quite a generous assumption because most of bees, they won't live beyond 30 or 32 days. So let's say 6 weeks for in this case. So for each individual bee that is in June, July, August, or early September, they only taking 127.4 nanogram in their lifetime. I hope that most of the audience follow me up to this point. So in order to calculate the dose, you have to assume some numbers that may or may not be correct, which is highlighted in the red taxes. Is that really 50,000 B? We don't know. It could be 45,000. It could be 55,000. But as a rule of the thumb, 50,000 B in that hive is a good estimate, especially for the healthy, strong hive. Does each bee really live longer than 6 weeks? We don't know. It's very difficult, if not possible, to count how many days that individual bee in the hive they live. So we would not publish those numbers based on some estimation parameters. But one number that for sure that 100% accurate is this 400 microgram per kilogram of high fructose corn syrup per week. This is why we publish. Obviously, those people that criticize our study, their mathematical capability is probably worse than elementary school students. And that's what I conclude with. So how much is 127 nanogram per bee's lifetime? It's only 0.2% of a grain of salt. Anybody try to weigh out 127 nanogram of something in the lab is a waste of effort because this weight is way, way, way too small. It cannot be weighted in most of the balances in the laboratory. And that's why we only report 400. 400, again, as I mentioned over and over again, is the number that we can live with. We can guarantee, we can testify. This is the exact number. But in terms of calculating down to the individual bee's daily dosage, there are some estimation. That's why we don't report this in the paper. But those people that criticize our study use this as their loudest criticism, which I found very ironic and feel sorry for their mathematical capabilities. So if you look at 20 instead of 40 microgram per kilogram of the sugar water, at the lowest dosage, which is 20 times lower than the 400 dosage, we have 100% mortality. That is equivalent to 0.14 nanogram per bee per day. Let me repeat this, 0.14 nanogram per bee per day. So why we don't report this dosage instead of 20? I explained the situation, the reason, and our reasoning in the previous slide. So we actually repeat the study, and we have the same result in the following years. But in the following years, we look into this phenomenon even further. So what happened to this hive? in early summers, in July 2010, this was a very healthy and strong hive. At the time, I would say most of the beekeeper would expect this hive be overwinter successfully, safely, without any problem. But a couple months later, once the winter was over, the left-hand side beehive become the picture of the right-hand side. So from the beekeeping practice, this is shocking, horrifying. From the scientific research perspective, this could be quite interesting because we can look into those photos and then try to come up with something that we can explain this. So here is what we come up with, the most plausible explanation of colony collapse disorder. We come up with this terminology called transgenerational effect of sublethal neonicotinoid exposure to honeybees. Every single term is very important here. So transgenerational is important. Sublethal is very important. Obviously, it has to be neonicotinoid on bees. So what we're talking about here, if you look at F0, this generation, this is the bee package that we purchased from the vendor. We start build up the hive from the very beginning. We don't use any leftover beehive because we are concerned about the previous exposure to neonicotinoid. After all, neonicotinoid are very popular insecticides. We knew that. So F0 is the generation that we are started to build upon. So that's March. And then by the time that we reach to the F3, they are strong enough. The hive is full of adult bees. So we start the dosing regimen. So that's the beginning of July. So F3, F4, and F5 were the three generations that those bees has the luxury to take some sugar water that come with a little bit of neonicotinoid residues. But none of them die between July and September. So obviously, the dosage that we gave to the bee in the sugar water were not high enough to trigger acute mortalities. So this is very different to pesticide that cause a lot of bee deaths on site because the high concentration. The concentrations that we use here were low enough that is safe for the bee during that dosing regimen. F6 was the generation that we stopped prepare the hive for the overwinters. So we remove pesticide sugar water at the end of the September. So October and November were the months that the beehive built up themselves, getting ready to overwinter. Also, none of the hive died between up to the November month. Okay, F7 and beyond were the generation that never took a sip of the pesticide contained sugar water, but they were the generation that suffered from CCDs, started from December 2010. So if we discontinue or end the experiment at the end of F5 generation because nothing happened, then we lost tons of information. This is also true if we terminate the experiment at the end of F6 generation because nothing happened to those hives. But if we stay with the experiment longer, by the time the winter was over, we look at this F7 generations, we found 15 out of 16 hives empty. So how to explain this phenomenon? Again, we believe that because of the sublethal exposures, we actually make the bee passing along the toxicities from one generation to the later generation and later generations. So this is where the transgenerational phenomenon came from. If you look at those mortality symptoms at the end, if you look at the end of experiment from different treatment and different outcome, this is the CCD on the upper left-hand side. This is the CCD hive. On the upper right-hand side, this is the control hive, that of disease. At the bottom of this hive, the picture, bottom of the picture, this hive actually is a control, was a control hive, but they overwinter successfully, and we can see there's still a lot of adult bee left. So if you make a comparison, the triangle comparison, it won't be too difficult to find out that pesticide probably is the most prominent answer to colony clot disorder. So I want to use this information that published way earlier in 1970 as a reason to support our explanation. So this cross-sectional view of the winter colony cluster is a perfect explanation of why honeybee as a whole, as a hive, can over the winter, and also to highlight how temperature or energy metabolism is critical for the beehive to be able to overwinter. So those data in terms of a temperature reading were taken on February 25th, probably somewhere earlier than 1971. 5 o'clock in the afternoon, when the ambient temperature was minus 11 degrees Celsius, okay, which is very cold. Inside the hive, in the area there's no bee cluster there, temperature was minus 7 degrees. But when the cluster, if you measure the temperature in the different layer of the cluster, the temperature actually rise from 7 degrees Celsius to all the way in the middle of the cluster. There was 33 degrees Celsius. So I have to mention this before we go further on these pictures. Before the winter arrived, healthy hive, all the bees in this healthy hive would actually travel down to the bottom of the hive. Because the winter, the low temperature is very cold, bee, instead of scattered around, they actually form this cluster, okay? When outside temperature is getting colder and colder and colder, the cluster is getting tighter and tighter and tighter. This is universal reaction to many organisms when the winter, when the low temperature happen in the ambient environment. Human being, because we live in the air conditioned housing, we can turn on the heat very easily and quickly we can reach to a very comfortable temperatures. But in the wilderness, the organism doesn't do it this way. That's why some bigger organisms like bear, for example, they went into hibernation in the winter because they need to maintain their basic or baseline energy metabolism. But honeybee, they form the cluster. So the cluster was at the bottom of the hive when the winter first arrived. And then the cluster slowly move vertically up. And in the path of moving those vertically, the heat that generate from moving the wings of the individual bees not only generate enough temperature to keep everybody warm and safe, the escaping temperature or the heat could melt a little bit of the honey so they can consume. At minus degrees Celsius inside the hive, honey were in a very solid state, like a cube of ice, right? But the beehive, the cluster, would able to melt those hive because the heat only go to the upper direction, not to the side or to the bottom. Again, this is a wonderful evolution of bee. Otherwise, they won't be able to do it this way. So by the time they reach to the top of the hive, hopefully the winter was over and they exhaust all the stored honey for the winter, and the bee can go out and start their pollination activity again on a yearly basis. So this picture tells us that energy metabolism is very critical because they need to maintain the right temperature. So the question is why in the middle of the cluster, the black spot, they need to be 33 degrees Celsius. I believe this, some of the audience will probably make a guess that this is where the queen lived. The answer is correct. By the time the cluster moved to the top of the hive, the queen sensed the spring is coming, or winter was about to over and the spring is not too far. She started laying egg to increase the size of the bees. Otherwise, they couldn't start this pollination in the right number of the bees, right? For the safety of the queen and the egg, the area has to be 33 degrees Celsius. There's no negotiation. And that's why if you think about this, 33 degrees Celsius versus -7 versus -11 outside the hive, bee has to work so hard to keep the temperature this high. Again, if there is an energy metabolism problem in the bees, then I don't believe that they can generate enough heat to maintain 33 degree, this kind of elevated level, right? So go back to the picture that I just showed you in the earlier presentation. Look at the middle part of this frame on the upper left hand side. The cluster was there, agree, right? But the cluster is not big enough. And the cluster probably collapsed earlier before the winter was over. But if you look at the picture on the lower right hand side, the cluster where the bees were much larger than the CCD hive, and we still have enough bees at the end of the winter so they can start take on the pollination activity, right? So if you look at these two photos, obviously it tells you that cluster did happen, take shape, but didn't last long enough to over the winter, and then the colony collapsed prior to the arrival of spring. So look at these pictures, a series of pictures. This is how we determine how many dead bees that you can collect from the hive. It's almost impossible to count one after another one because there's just simply too many dead bees. This is the dead colony from the control group. We actually measure 3.5 liters of the dead bees. Obviously, most of them are here. But when we try to determine how many dead bees we can collect from CCD hive, we can only collect 0.2 liters of those dead bees. So the question is, where were the other bees? Where were those 30 or 40,000 dead bees? We cannot account it for them. So we keep asking questions about these pictures. And we come up with something that is plausible and reasonable, which is the mitochondria. We know the energy metabolism is very important. We know generating heat to maintain ideal temperature inside a hive when winter were there is very important. So what else we can come up with other than mitochondria, right? So if you look at this slide, this is honeybees mitochondria. It's very different to nucleus DNA. It's a circle shape. It's only have 22 RNA encoded genes. There's about 13 protein encoded genes. So comparing to nucleus DNA, mitochondrial DNA is very simple and very straightforward. Mitochondria in honeybees were the same as human, believe it or not. Mitochondria is very important in terms of energy production via the ATP cycles or oxidation. This is also true in humans. It has the same base pair, 16,500. This is also true in human. Here is very important. Mitochondrial DNA doesn't matter is a good DNA or damage. It all come from the maternal side. The paternal side has very little to do with our mitochondrial DNAs. Mitochondrial DNA, we know, is very sensitive or susceptible to environmental insult than nuclear DNA. This is also true in human. And we also know that 85%, just because they only have a few, right, 20 something. So we know that most of the mitochondrial DNA are functional DNA. So if we damage those mitochondrial DNA, the consequence is very severe because we're damaging functional DNAs. One thing I should actually emphasize here is that mitochondrial DNA, once damaged, they don't repair. This is very different to nuclear DNA. So what they're gonna do, they actually replicate all the DNAs, both good and damaged. And they hope, we hope, or mitochondrial DNA hope, that by replicating good DNAs, the mitochondria could still function. But at the time, they also replicate damaged DNAs, right? So over the time, if the damage is persistent or ongoing, then we reach the threshold that DNA, the mitochondria has way too many damaged DNA, and this mitochondria probably gonna lose the function and then trigger the cell death. So in our following studies, we come up with a little bit different study design in terms of we collect bees before the dosing, during dosing, and right after dosing. This is what we do. What we try to do is to identify generations, different generation of bee, although they are in the same hive, see how their mitochondrial DNA respond to pre, during, and post neonicotinoid dosing. So keep in mind that those are the different bees from different brood generations, but they were in the same hive. They're from the same queens. This is what we come up with. We saw increasing mitochondrial DNA copy number, which echoing the increasing damage to mitochondrial DNA. In different generation of honeybees, as a result of the neonicotinoid dosing, we use two different neonicotinoids. One is imidacloprid, the other one is clothianidin. If you look at the photo, the figure A on your left-hand side, the gray line or gray data set representing the control bees, the red and the green representing neonicotinoid exposed bees. You can see that before the dosing, the mitochondrial DNA in different type of bee, they're essentially the same. But during the exposures, we start seeing the deviations from the control. And then after the exposures, the deviation is even more obvious, right? So the question is why we also see the rise of a mitochondrial DNA damage in control hive in the following generation without pesticide exposures. Well, obviously, the increase of mitochondrial DNA damage in the control bee representing agent. We know that agent is corresponding to the quality of your mitochondria. And nowadays we use mitochondria as a checkpoint in terms of for the agent problems. And this study actually able to reflect the agent of the queen herself and her offspring daughters in this case. So why mitochondrial DNA damage is very important or significant? Because it actually demonstrates our initial hypothesis, which is the existence of transgenerational effect of sublethal neonicotinoid exposures. This data set, they indirectly explain why bees disappear in winter following the sublethal neonicotinoid exposures. And the cause of the bee death is not acute or by pathogen infestation. Otherwise, we're going to see a lot of dead bees, right? But likely in the form of progressive energy metabolism decline because of the cumulative mitochondrial DNA damage. This is very plausible in our studies. So how significant mitochondrial DNA damage is to the disease burden in the population? We know that mitochondrial DNA damage is associated with various diseases. The most obvious ones are cancers, neurodevelopmental and neurodegenerative disease. So the data set that I just presented to you was published last year in April and in environmental science and technology letters. And our study was selected as the cover study for that issue just because of the novelty of the data set and also the study linked to a very serious environmental problem that is still ongoing right now. So before we conclude this presentation, I want to propose another very bold hypothesis. Although we can see that is happening right now in our study, is that everybody heard or understand this phenomenon, which is called only dose produce the toxic effect, or everything is toxic, depends upon the dosage. So this is sort of like people understand the meaning of the toxicities, right? But if you look at these two paragraphs, there's no time associated with either toxic exposure or the disease or the dosing. So based on our honeybee study, we believe that dose is important as well as the length of exposures. Because if you look at honeybee, the study, the F3, F4, F5, they were fine, meaning that dosage is fine. By the classic definition, they are by Paracelsus, right? But if we wait until F7 or F8, when we extend the time, obviously the previous dosage that we thought safe were not safe because they trigger CCDs. This is why this come very important. This concept come very important to the overall public health research is called precautionary principles. We learned this precautionary principle much earlier because we know this phrase again, canary in the coal mine. Why canary in the coal mine matter to public health? Because this is the first public health knowledge in terms of how important prevention is. Way back in the 19th centuries, when miners, they go down to the mine to work, they found that some of their partners were dead in the coal mine. They don't know what happens. There's no real-time monitoring. There's no research associated with mining and so on and so forth. But mortality among the mining workers happen all the time. So those miners become smart enough. Next time when they go down to the mine, they carry a bird cage with 2 live canary in it. If the bird becomes struggling way below the mine, then the miner would leave the mine and then come out right away because the bird was struggling, right? This is the first prevention measure that we know that document in the history. You can still see this empty bird cage in the Kentucky Historical Society Museum. And then 60 years ago, this book published by Rachel Carson, Silent Spring, which also served as the prevention purpose. She sound the alert of no bird singing in the winter because she found many dead birds, eggshells, and unborn bird has DDT in it. So Silent Spring is a modern era of the precautionary principle. Now we have these CCDs. bee has no knowledge of where the flower has been sprayed with neonicotinoid. They still go there and then collect the bees, the nectar and the pollen, and then couple brood generation later during the winter, when they found out that those a little bit of neonicotinoid can make their energy metabolism problematic. They cannot generate enough heat to maintain the right temperatures inside the hive, the whole colony collapse. This is another type of silent spring, except the culprit is not bird, now it's a bee, but the causing agent is the same, pesticides. It's also the outcome is the same. There's the lack of the bee population. So the question is how many type of organism in the wilderness they suffer the same degree of the pesticide toxicities, but we just don't know yet. In this case, because of the Hackenberg, he is the commercial beekeeper. He sounded alert because of the bee loss in the massive numbers, and then he called the government. But not every single wild organism, they actually were raised by individuals. So they are losing the size of their population in a gradual manner without the notice of the human beings. So now we are looking at this honeybee population declining. We know that this is the cause of these pesticides. We need to be serious about whether those neonicotinoids should be used in the society. So this is why this research becomes very important to maintaining a healthy and a very good ecological environment. Thank you for your attention. This is the end of my presentation.